Um, so yeah, as Mariska was saying, I'm gonna talk a bit about development interactions in the US community. Yes, you already gave a very nice intro, so I can skip most of that. I would say I started about 5 years ago at MSK in Nikki Schultz's lab. So cBioPortal started about a decade ago, and Nikki and Ethan, they were the founders of cBioPortal, and I kind of got on board when cBioPortal really chose to go full open source. And yeah, I've been dealing a lot with like trying to scale, it got so popular. So the developer, that we got more developers, the community grew, there were more users. So all kinds of problems kind of related to scaling that. And so for this presentation, I'm going to talk a bit about, the community, how we manage the community. So I think Sjoerd touched a few of these points, so I can go through those pretty quickly. And then talk a bit about recent developments, so like new features and other things that we've done. And I'm going to try to give some idea of like our future plans and what we're developing. And obviously the list is like endless, but I'll try to sort of show what we're planning. So yeah, first all the faces. So these are, all the people working on cBioPortal. Yeah, there are many more, like, not everybody fits on here, but there are also others, that maybe contribute once, in several years. But I would say like most of these people, they contribute, at least once a month or so in capacity or another. And so here is a listing of all the different institutions and names. So we see MSK, Dana-Farber, CHOP. So yeah, Sjoerd sort of mentioned this already, but so the US community is really this part, these first two columns here, and we're all on the East Coast. So, I mean, I would say that Princess Margaret, they're also very integrated in kind of our developers, like our development team, because they're on the same time zone, so it's a little easier. And then, yeah, here you can see all the funders. So it's like a wide variety of different funds that come in and different features that people would like to see developed. And here's a list of different people that have worked on cBioPortal before. So, even though the community has grown a lot and, a lot has happened in the last years, the goal has always kind of been the same, to really make, complex cancer genomic data accessible and interpretable. And that's really what we try to stick to, whenever we work on something. And that's kind of what the whole community and also like this day is all about. And so one way, like the accessible part, we have various data sources that go into cBioPortal. So there's a whole curation team. So several people that work on looking at the papers, getting the data, and putting it into cBioPortal.org, the public website. These can sometimes be user requests. Sometimes it comes from from someone in the community. Yeah, the Cancer Genome Atlas in there, various other, the Cancer Cell Line is, Sjoerd showed that as well. MSK Impact, so this is a big, basically the assay that's being done at MSKCC. Yeah, at our institute, they've organized it in a way that anybody at MSK can access this data set. So it's like a panel of like 400 or so genes. And right now about 50,000 or so patients have been sequenced. And the public one, we currently only have the published study, which is about 10,000. So that one you can access there. But I think that's been really cool at MSK, that they've been able to kind of say, all right, we're just going to make this data set public to anyone at MSK, and then just have clear publication guidelines, and then anybody is able to use it. And then there's also the AACR Project Genie, of which NKI is also part, and several other institutions to combine some of this molecular profiling into one cBioPortal instance, and that one's access controlled. So you see a combination of data. People kind of went over this already. Yeah, the other layer that cBioPortal adds is this interpretability. So really combining other resources that are already available, like PFAM and PDB and UniProt, to kind of get the background biological data, ways to kind of interpret variants, so kind of clinical implications of variants on KB and CIVIC, and several other several other sources for variant prevalence and predicted functional effects. So those are kind of the two main sides of cBioPortal, and so it's used a lot. There's around 4,000 daily visits, so you can see over the years how it's increased, and the number of citations, and like in the last year there were over a million visits, and this is just like the public instances. And here's a map of like all like the ones that we know of, the different local installations. I'm sure there's many more, but you can see, it's a combination of hospitals, like academic institutions, government institutions, and commercial companies. So a wide variety. So overall, I hope you sort of see, through these slides that, the community is quite big. There's like a lot of different funding sources, and the user base is broad. And so overall, it looks like a very healthy community. Yeah, so a bit about the community management. So obviously, with this size community, there's a lot of maintenance kind of involved. So we have this mailing list. So there's, users of the website and kind of instance maintainers that ask questions on there. And this is really a community effort to manage. So also the Hive is also managing this, and we just have like a weekly rotating schedule of somebody taking care of the mailing list and making sure that all questions are answered. So that's cool that this has really become like something that everybody contributes to. And I think that's very important, the focus on the users really. And that's why it's good that everybody in the community is involved. And then in terms of code, so, obviously somebody has to look at the bug reports. If somebody sends in new features, people have to review it. Like Sjoerd mentioned, sometimes, people submit requests for comments for like big new features to kind of describe an implementation plan. So that has to be reviewed. So I would say like, yeah, a lot of this kind of happens still mostly in the US community. So this might be something also to further grow in the NL community. Yeah, then there's a maintenance of the curated data, so updates to the data, fixes, etc. Then there's the maintenance of the servers. So we mostly maintain the servers at MSK, but we do share a lot of how we are deploying our servers, and that's a bit of a community effort as well, where we kind of share how we're doing authentication and all that stuff. So we have a bi-weekly community meeting where people are presenting features, and then we have a steering meeting weekly where we discuss things that people see in the mailing list, but also sort of conferences that are coming up or things like that, or ideas for new features. And then we use Slack a lot to kind of have asynchronous chatting and just interaction between the community. And yeah, so over the last years, we've really grown out this a lot as well, kind of the development management, because the team grew, and we have, of course, like teams in different areas of the world working in different timelines. So kind of having a clear way of managing all the work that's being done, that helped a lot. So on our end, we're using the Scrum a lot. So we have in MSK, we have like 3 sort of development teams, and also the other institutions are embedded in that. So their developers are often also on our planning meetings and stand-up meetings. And yeah, that works pretty well since they're in the same time zone. It's a little trickier, obviously, you know, with Europe, but it might make sense at some point to have something similar in a European time zone. And we use continuous integration a lot. So this testing that Stuart also showed to make sure that things don't break. So Stuart was also showing some features like the gene sets. So we don't really have that data, but then they build in testing to make sure that we don't accidentally break it. So that's kind of the way you can make sure you get your features merged into the main branch, but we don't have to kind of manually test to make sure that we don't break anybody's features. And so, yeah, you can see here we use GitHub a lot. You have an issue tracker. The developer, implements a feature, fixes a bug, then they send this pull request that gets reviewed, and maybe they have to make some changes. We have the testing that goes off. We also do what is called product review. So usually people that are intimately familiar with the cBioPortal product, they test it just to make sure that everything behaves as expected. So some sort of user testing, and then we can release to the cloud, and we're deploying to production multiple times per day, and that was like a few years ago much harder, and now that's working pretty well. And that's, I think, in large part also due to the automated testing that we can actually do this. So in recent developments, so there are so many new features that have been developed in the past 2 years. So this is just some of them. I'll show a few slides around that. I just wanted to show, so we have I just said we have, let's see, like daily deployments to production. We also do weekly community releases. So every week, anybody can just pull the code and use the latest version, and we kind of describe each week what has happened. So this is quite detailed, and you can look into that more. And yeah, so in terms of the new, and then we also did this webinar series to which Stuart alluded, which I highly recommend to watch if you just want to get more familiar with how to use cBioPortal. So it's like 5 episodes, and every one is like 45 minutes. And yeah, I think they're really, if you watch them, you'll have a really good idea of how to use it and how useful it is. So yeah, the user interaction flow of cBioPortal, there's really kind of 3 main pages. You have the query results view, where when you search for a specific gene, you go to this page. You have the study view to kind of get an overview of the cohort, and then we have the patient view, and some of these have been shown. And recently we also added the group comparison. So that's really a big new main page, which allows you to kind of compare different cohorts. So you can select something on the study view, a particular cancer with particular mutations, and you compare it against another one. And you can compare survival, mutations, all types of molecular profiles and clinical data. And yeah, this is what somebody requested. So there are specific features for cfDNA visualization. So as you can see here, this is on the patient view. The blue samples here are cfDNA samples, and you can see them right here, and kind of how this AKT1 mutation, how the allele frequency changes over time. So this is one of the features that we've built out around cfDNA, and you see also some light circles here, and if you mouse over those, it will show you the number of supporting reads. So if you're familiar with mutation calling, the dark ones are really called, and the light ones haven't really been called, but there are some supporting reads for that mutation, which is very useful in ctDNA analysis. Yeah, we've done a lot for AACR, Genie. We've done a lot of improvements on longitudinal clinical data, and so we've made some improvements on the timeline. And, you know, when you will use a new version, you will have all this, but we have added some color coding, for instance, for imaging assessment. So you can see if the cancer is progressing or if it's stable, things like that. We also have improved cohort selection. So sort of, you know, we have here, you see when the sample is collected and whether it was before a certain treatment or after a certain treatment. So on the study view, you can now select, for instance, samples that were collected before a certain treatment and compare them, you know, against something that's after that particular treatment. And so if you already have this timeline data, it will automatically use that to kind of make these charts on the study view to allow you to make these types of selections. And there's many more things that we're working on and that we've worked on, but just what kind of wanted to keep it to that and pick those two, mostly because they were mentioned before. Yeah, so future plans, we have, you know, like I said, the longitudinal clinical data we'll probably work on more. Yeah, whole genomes, structural variants, we're working on that a lot to improve that. Stuart mentioned single cell, that's also something we're looking into and how to incorporate that in cBioPortal. Yeah, mutational signatures, immuno-oncology data support, yeah, copy number, specific, sorry, so this is FACET, so it's basically for cancer cell fraction and allele-specific copy number support. And we also want to work more on doing kind of similar patients recommendations. So if you look at a patient, that it will tell you, oh, this patient is very similar to this other patient, maybe you want to take a look at that. And then just overall better performance and improve the data importing. So that's pretty much it.
Perfect. Thank you, Ino. The timer just went off, so that's like a well-done egg. So the program says that right now we're actually preparing for the breakout, but there is one question that I would allow before we are breaking out. And that is, how many institutions, academic centers in the US are using cBioPortal? And do they have their own local server, or are they all connected to the MSK server? What is your insight into this, Ino?
So I don't know the exact number. There are many, and everybody has their local instance. So that's the answer.
Yes, indeed. But there are also more than 300 studies in cBioPortal public, and AACR Genie contains data from 19 institutes all over the world with more than 100K samples. So that gives a reference on that. So thank you, Ino, for the lovely presentation and the inclusion of the cfDNA slides.